What I was planning to talk about today is really a simple process of the cell spreading onto a surface and sensing the rigidity of that surface. And it's a basic fundamental process, and what I feel we can gather from looking at that in detail is really the sort of thought process that the cell goes through, for want of a better term, in approaching a matrix. Obviously, the microenvironment of the cell determines its behavior, and the inappropriate response to that microenvironment leads to cancer, leads to aging, or we think it's inappropriate, at least if we're aging like me, and other types of behavior during development as well. So it's a critical issue to understand how the cell really interacts with its environment, and that's what I'll be focusing on today. The issue in general is, how do you make all these organs and this body from single cells, and how do those single cells sense where they are in this body and react appropriately, grow when they need to grow for regeneration, but don't grow when they shouldn't grow, i.e. when cancer happens. So it's obviously an important issue in terms of how the cell reacts to its environment. And what I'll be focusing on, at least in part of my lecture today, is the idea that one of the major ways or major themes throughout various levels of mechanosensing is the sarcomeric unit, which has alpha-actinin at the ends anchoring these actin filaments, myosin in the middle pulling anti-parallel actin, and contractile forces are indeed the major forces that cells generate, and we'll say that they are, or at least I'll posit today, that they are the way in which cells sense the rigidity of their environment. Okay. Many have talked about actin polymerization forces. However, those do not seem to account for most, if not all, of the large force generating that occurs within the system. So movement of fibers, movement of cells is really driven by contractile forces, although the assembly of actin needed for that contraction is due to the actin polymerization. One of the major aspects that I want to emphasize today is really the scale of the process that I'll be talking about. In considering mechanical phenomena, we know that geometry and force sensing, all of which can occur in cells, can occur very rapidly. So you tug on a cell, its response is within a fraction of a second typically, maybe up to 5 seconds if it's a slow issue. The response to curvature, likewise, this response to distribution of matrix, likewise, and that then leads to a transduction signal, which is either calcium entry, if you believe in that, a stretching of a protein, which leads to a small G protein activation, and a diffusion of that signal throughout the cytoplasm. Then there's a response. The G protein binds to its effectors, calcium binds to calmodulin, causes a change in a protein, and that leads to actin assembly or myosin contraction, which changes the original condition. So this is a cycle. Okay? And when we look at cells after several days in culture or several hours in culture at the minimum, typically, hundreds to thousands of those cycles have gone on. So if we talk about a protein affecting a process, it's really and we use a final morphology of the cell as our assay. We don't know where in that series of 1,000 cycles that protein is important. Okay. So if we wish to understand how this system works, looking at the end is a very poor way of doing so. Okay. It's sort of like asking, why someone doesn't reach the end of a 1,000-mile journey when you know they've had to take boats, trains, planes, etc., automobiles to get there. Okay. So you don't know at which step the system fails. Okay. So, and what I'm arguing for today is the idea that there are a set number of such processes, motile processes within cells. They typically behave in a standard way. And so if we understand each one of those 20 or 30 different processes, we understand how the cell can behave, and then it becomes a question of which of these standard types of motility is used under which circumstances. Okay. So as opposed to the bioinformatician approach, which is to say that there's 6,000 proteins within the cell, and the concentration of all 6,000 of those is a critical determinant of how that cell behaves, I'm saying it's a lot simpler problem. If we take the view that there are a certain set of standard behaviors, and once we define those, then we can understand how that system works. The example that I can give that says that the system is indeed deterministic, even though it's based upon stochastic processes, is just the simple one of looking at two identical twins. Those two identical twins have grown and have developed independently, and there are a whole set of decisions that were made by the cells independently, and yet they came to pretty much the same conclusion in the end, and often you can't distinguish between them. So that means that there is a deterministic element. And we think that the process of these cycles is critical for determining that. So there must be a way of integration in these systems. So what we're really focusing on here is not the components. We understand what the components are of the cell. We understand how many of those interact with one another or how actin filaments assemble. We can see how cells move in bulk. And we can use the analogy of an automobile. What we understand relatively little is the subcellular machines, so to speak, that can be likened to this automobile engine. The process that I'll be talking about today is this periodic contraction that we observed some time ago of the cell edge as the cell is spreading. This occurs in roughly a 2-micron region, contains thousands of components, and can be likened to this automobile engine in several respects. One, if you take the parts of this engine and lay them out on the table or on the floor here, you'd be hard put to understand how those parts actually help to make that engine work. Two, that in this system and in this system, timing, position, forces are critical to make it work. Okay. It's not just the components, but it's how those components interact in a dynamic fashion that makes it work. Further, this can be placed in a variety of different car bodies, and it can work just fine. And so this activity we see in fibroblasts, in endothelial cells, something similar happens in growth cones and epithelial cells as well. So these modular or emergent properties, or however you wish to describe them, can be considered as a unit or a tool that can be applied in a variety of different cell backgrounds with the same generalizable outcome. Okay. And that potentially simplifies the overall process that we have to consider, namely, how does a cell do something? Okay. And so that if it relies upon standard activities, then we only need to understand those important ones. We collaborated for a while with Nam Su, who was here at MIT before, now is president of KAIST. And he was involved in engineering robust devices. He was responsible for building a better flip top for beer cans and for making a space docking module as well, using pretty much the same principles. Okay. And the principles that are applied to those robust devices also apply to cells. So the leading issue is compartmentalization of functions. They're localized. Okay. If I go back to this region of the cell is a localized region which is specialized for this periodic contraction. It's not a membrane-bounded compartment, but it's a compartment nonetheless. And it's a functional compartment in that system. They're modular components. Indeed, they're modules for actin assembly, for myosin contraction, etc., for adhesions. Functions are largely digital, not analog. So the way the cell produces the right amount of mRNA so that it can produce proteins is not by continuously producing mRNA and adjusting by a rheostat the rate of that production, but indeed turns on, produces a burst of it, turns off, and waits for another signal to turn that on. Okay. This leads into the next issue of term limits. In the Bush era, at least, we did not want that to continue, many of us at least. And it was good that there was a term limit built in. The same sort of thinking applies to cells as well. They don't wish to go down the wrong path for very long before they, are forced to make the decision again of whether they'd wish to move. So indeed, the default pathway or the default activity is for systems to turn off. And so if you look at the motility seems to be a single exponential decay in terms of its lifetime. So that built into this system is an automatic turnoff. That applies to many biological systems. Okay. And then you have to have an active signal to turn it on, and that's the way you regulate, okay, a robust device. Cycles, we'll talk a lot about cycles today. Some of these are futile, but futile cycles of phosphorylation, dephosphorylation, stretching of proteins, and relaxation are not necessarily futile. And we think that they, or at least we'll posit, that they are part of this integration process that must occur to make a stochastic process into a deterministic process. Okay. So, and then there are several variations of important functions, namely that you find out if you knock out many proteins, the cells will go on just fine, or you can knock out a given function and an alternative function will come into play. And finally, there are often phases or linked functions with sharp transitions. We'll see some of those today in the cell spreading onto the surface, and totally different protein activities and protein processes are active in one phase versus another, and the transitions from one phase to another occur within seconds. Another way to think about this is that your response to environmental stimuli is dependent upon your phase in the day. Obviously, at 2 a.m., you will respond quite differently to the same stimulus that you have at this time, at least hopefully. Okay. So, indeed, the same thing happens with cells, that they will respond differently, i.e. are in different states at different times. So going into particulars, what I'm going to be focusing on a lot is cell spreading and looking in detail at the steps leading to this periodic contraction and then to adhesion assembly afterwards. So the cell is extending here through actin assembly at the leading edge. It pulls, and when it pulls, it sets down a new adhesion site, a concentration of the beta-3 integrin, and there's another concentration here, and we find that myosin is localized behind this second adhesion site, and it's pulling on the actin, condensing that into circumferential bands of actin, which can go on to become stress fibers. Then, once it pulls that actin away, new actin starts to polymerize at the leading edge in this upper layer. Recent studies from Jiawei Zhang show very nicely the 2 layers of actin within this region. And as it polymerizes, this new myosin will assemble between these sites, and this implies a anti-parallel arrangement of actin, which we'll talk about more, i.e. similar to a sarcomere. And then when that actin reaches that myosin, you get another pull and the cycle repeats. The bending that occurs here can be simulated by these 2 post-it notes, in that the upper layer of actin gets pulled, the lower layer is anchored, and so you get a bending occurring. Okay. Please don't hesitate to interrupt me with questions as we go along. You know, all of you have different backgrounds, and so, but if you have a question, it's likely that your neighbor has a question similar. So don't hesitate to interrupt. Now, the issue then becomes, what are the long-term effects of these tests of the environment? And Adam Engler and Dennis Disher's lab looked at this some time ago using a collagen substrate on a elastic background polyacrylamide. There are some doubts raised by work from Joachim Spatz and Viola Vogel as to whether or not there is some change in the architecture as you go to these low rigidity polyacrylamide surfaces, but be that as it may, the final result is pretty much the same, namely that the cells respond differently. This is 4 hours and 4 days to the rigidity of the environment and the same presumed chemistry in terms of collagen, and such that at low rigidity they become neuron-like, at intermediate they become muscle-like, and at high they become bone-like in terms of their differentiation. So this is a long-term behavior, but even at earliest times, we can see that the cell senses the rigidity of the environment. So here we're putting mesenchymal stem cells onto soft or rigid pillars and looking at their initial reactions to those surfaces. And we see that when they spread on the soft, i.e. tall pillars versus the short pillars, same diameter, they find that they become more dendritic here, more quote-unquote uniform spreading here, so that the process of rigidity sensing can occur very rapidly. It doesn't require the 4 hours or 4 days that were used in those previous studies. Okay. So that says then that the process by which the cells sense or can sense rigidity occurs in a rapid fashion. So we'll look at this from the point of view of what are the initial steps of the interaction with the surface. We can control that. We know when we start that interaction, and so we can break the process down into a series of steps. That will be testing the surface matrix, and I'll emphasize clustering there. There'll be rapid spreading without contraction, at least in this isotropic spreading case. Contractile spreading, this process that I talked about of the periodic contractions, which tests the rigidity of the surface. Then adhesions mature with further actin contraction, and you have continued testing of the surface over time, i.e. through the adhesions. So looking at the multimeric nature or clustering nature of the initial interactions, we see that a fibronectin trimer, and I'll describe what that is in a moment, binds quite differently to cells than does a fibronectin monomer. So they're simply added in the solution, and there's a rapid wash and fixation at the same time, and then visualization afterwards. Okay. And what you find is that the trimer is binding at the edge, whereas the monomer is binding over the whole surface. The composition of that trimer was essentially using a hexabrachian or tenacin trimerization domain coupled to a tenacin spacer arm and then to the fibronectin 7 to 10 domain, which has the RGD loop here. This was a construct provided by Harold Erickson, while I was still at Duke. And so they're by and large the same, except for this trimerization domain. So drawn to scale here is the alpha-beta integrin dimer, about 20 nanometers out from the surface. This is about 60 nanometers in width, so that we can see how large this complex is. We went on to use beads in laser tweezers at low concentrations of fibronectin trimer on the surface, so that we were presumably looking at single molecule interactions. And what we found with the trimer and not with the monomer was that there was a Talin dependent 2 piconewton slip bond formed with the actin cytoskeleton. In other words, this actin cytoskeleton continues to move inward at a pretty steady pace of about 60 nanometers per second. And that couples with activated integrins, and you see it start to move, and then it pops back, moves and pops back. And so that we call the slip bond. Our good friend Evan Evans went back over the data and confirmed that it behaved as a slip bond. It's nice to have friends check your data for you. And what happens in general, if you have a more rigid trap, more rigid laser tweezers, you find that in a counterintuitive type of fashion, you're more often going to have a permanent attachment to the cytoskeleton, and the bead gets pulled out of the trap. Whereas at low rigidity traps, you find that you're more likely to break the attachment to the cytoskeleton, and it's less likely to be reinforced, indicating that there is a rigidity-dependent step in that overall process. Okay. Now, in the meantime, Joachim Spatz's lab has been producing surfaces with the diblock copolymers around gold, so that they can produce arrays of 5 to 6 nanometer gold dots spaced by arbitrary, or by defined, distances from one another, dependent upon the size of the diblock copolymer micelle. And then you use reactive ion etching to remove the diblock copolymer, and you're left with an array of gold dots to which you can affix a biotinylated self-assembling monolayer, and then you can add avidin, and then you can add biotinylated RGD here, and the cells will spread on those if the dots are spaced by 60 nanometers or less from one another. But when they are greater than 60 nanometers, there's an abrupt loss of adhesion to that surface. 60 nanometers was intriguing to us because that was roughly the length of that fibronectin trimer. The arms in that trimer were about 30 nanometers in length. And so we collaborated with Mark Schwartzman, a graduate student in Shalom Wynn's lab. And Shalom was at previously IBM, e-beam writing, and so he was able to produce arrays of gold dots, again roughly 6 nanometers in diameter, spaced by arbitrary values and in different arrays because he was using an e-beam writer. And so now we could look at a dimer or trimer arrays and ask the question, would the cells spread on those? And we were quite disappointed to find that although the E-beam written hexagonal arrays would support cell spreading at 50, 60 nanometers, but not at 80 or 100, repeating the results from the Schmatz lab, that the trimers and the dimers would not. And so that raised the question of, is this a density issue? Because obviously there are many more dots per square micron and many more RGDs per square micron in these arrays than there are in these. So Mark went back and looked at this issue by taking heptameric arrays in this now hexagon plus a central dot, and spacing those now at various distances to decrease the overall density of dots per square micron. And when he started out at 216, that was essentially close packed. And down here at 51, these are separated by about 400 nanometers from one another. And the percent of cells that spread was the same in all of these cases. 51 dots per square micron was the density that we had for the dimeric arrays. And so that said that really the density of dots was not the issue. That we could have a cluster at that density, 50 dots per square microns, if they were clustered in these arrays, they would support spreading. So we went back and now produced at a constant number of dots per square micron, dimeric, trimeric, tetrameric, pentameric, hexameric, and heptameric arrays of dots. And asked the question, would the cells spread on those? And what we see here is that they, again, don't spread on the dimers and trimers, but do spread on the tetramers, pentamers, hexamers, and heptamers. Indicating that you need to have 3 or 4 gathered together in thy name to get a proper reaction. Yeah.
What is the y-axis percent of spread cells? Is that the percent of the population that spreads, or is that a...
That's the percentage of the population that spread after a half an hour. Okay. So, other questions here? So, what this is emphasizing is that you need to have a certain number of integrins, a minimum number of integrins of about 3 or 4, clustered within 60 nanometers to get an adhesion that will support the spreading of the cell. Okay. So we come up with a concept of a minimal adhesion unit here. Okay. And we're now looking at this again with these nano-patterned arrays to define what's different between a unit where the dots are 60 nanometers apart and a unit where they are 80 nanometers apart, which will not support spreading. So there obviously is a biochemical correlate to that. Now, looking at this from a different point of view, we've removed any rigidity from the surface by putting the same RGD construct attached to biotin onto lipids. And this is a supported lipid bilayer where everything is fluid here, and there is no barrier to diffusion except ones imposed by these lines here. These lines are nanofabricated, roughly 100 to 200 nanometer wide lines of chromium, which breaks the bilayer and you can't move beyond it, because if you look at it, do a photobleaching experiment, you find that the construct recovers with a lipid diffusion rate, 2 microns squared per second, but it doesn't recover in those regions which are blocked off by the lines, i.e. these square domains over here. So what now happens when cells are placed on this? What we see is that over time here, this is 25 seconds, we find that the RGD is being concentrated at the lines, as is paxillin, which is a cytoplasmic protein associated with adhesions. So that it seems these are being contracted. Okay. If the contraction is occurring, that means that you must have, or you should have, an actomyosin complex, harking back to the sarcomeric unit. And the way to test that is to now inhibit myosin contraction. Oh, one other point I forgot to bring up is that the growth of these clusters occurs independent of that actomyosin contraction. So we can inhibit actin polymerization, we can inhibit myosin, the clusters still grow, and they still accumulate cytoplasmic proteins such as talin, paxillin, and focal adhesion kinase in rough correspondence with the accumulation of RGD. And I've had an undergraduate student in the lab model this as a diffusion to a trap, and the data fit well with the known diffusion coefficients of integrins in the plane of the membrane diffusing to a trap. So that this clustering, initially at least, seems to be a property of the integrin activation that results in its clustering. Okay. When you think about it, it's a relatively profound issue because it explains quite simply why, when you have a uniform surface coated with a uniform distribution of fibronectin, you end up with adhesions where the integrins are concentrated in local regions. Okay. What it means is that there is an affinity in the lateral sense of integrins for integrins once they become activated. And that is sufficient to recruit many of these adhesion-dependent proteins. Vinculin, not. Vinculin doesn't come until you get contractile force applied to this. Okay. Any questions about this? So going back to the story, if we see these clusters and the clusters are moving to the lines, it should be myosin dependent, we're saying. And sure enough, when we add blebbistatin, a myosin 2 inhibitor, they don't move to the lines over time. They do assemble, however. This begs the question of where is the actin? Because you need anti-parallel arrays of actin for sarcomeric contraction. Indeed, we see that when these clusters form, actin polymerizes from those clusters. Okay. So this is sort of cute. What nature is doing is saying, you know, I need this minimal adhesion unit. I'm going to create this cluster. And the way I'm going to know that I have a cluster is by polymerizing actin from that cluster. So now that cluster can be contracted to other clusters by an actomyosin dependent manner. Okay. And this also begs the question of how do you get this actin polymerization? We could inhibit that with inhibitors of formin. And turns out one of the postdocs in the lab had worked on formins before and had constructs for FHOD1. And sure enough, at 5 minutes, you see a peak in the phosphorylation of FHOD1, i.e. cells in contact with the surface initially activate actin polymerization through FHOD1, and we find it at those clusters present, and there's a paper now that is in revision on this. So it seems that what's happening is that you have a cluster formation, you have the recruitment of FHOD1, a formin here, which is polymerizing actin from that site in a radial manner, and then myosin now will interact with that actin and actin from neighboring sites and will pull those together. What is the consequence of that pulling? Okay. If we have these barriers present, we find that 80% of the cells spread further. They activate a spreading response, whereas if the barriers are not present, less than 25% do. So what this supported bilayer enabled us to do was to uncover a new step, or previously unrecognized step, in the process of cell spreading. So what the cells are doing is, first of all, assembling a cluster, assembling actin from that cluster, pulling on those clusters now with myosin, and asking the question, does that matrix pull back? I.e., can I generate force? And if it's a supported bilayer and you don't generate force, you don't spread. But if you do generate force, you do spread. Okay. Any questions about that? So the cell is going through a series of if-then questions. Okay. It's not going to commit to spreading on a surface unless it knows that there's something on that surface it, one, binds to, and two, that's rigid, or at least not rigid in the sense of measuring the rigidity, but at least pulls back. Okay.
Excuse me.
Yeah.
So if there's no posts there, does the clusters form and this process happens and ends up in nothing, or?
The cell doesn't spread, it stays rounded, and eventually those disperse. Yeah.
So what initiates the formation of clustering?
The diffusion and trapping. So in other words, the integrin binds to RGD. This is now beta 3. It changes its conformation, and it has an affinity now for other RGD liganded beta 3 integrins in the lateral sense.
So if there is no posts, meaning no force, they form still, but the cell doesn't spread.
The cell doesn't spread. Okay? So the cell is asking and requiring, in a sense, that what it's binding to pull back. Okay. So consequently, it's not going to spread as a result of soluble fibronectin in serum, for example.
Like one thing that might not be too real clear is, let's say you're on a rigid surface, so you get binding to two RGDs on that surface. Now, you're saying that those would tend to merge, but if the RGDs are rigidly attached to the surface, they can't, right?
Right. So that's why there has to be a certain density of RGDs so that the RGDs are going to be close enough stochastically to bind integrins and form that initial cluster. Okay?
Okay. And I suppose you could even detach from, you could generate a force, detach from one of the RGDs, and then rebind to one that's closer, which would...
In theory, yeah. So, right.
Quick question about the difference between Paxillin and Vinculin.
Right.
Paxillin could accumulate without the internal machinery
Right.
generating force, so it just accumulates because there's integrins there binding to that structure?
That's sufficient, it seems, yeah.
Okay. But
Right.
then Vinculin requires...
Vinculin doesn't bind until you get the contraction, which is the cells spread here and then they contract. And when they contract, you see vinculin binding.
Okay, so is there like a conformational change, or is it...
We'll come back to that later on, but basically it correlates with the stretching of Talin, which has within it, embedded within the molecule, 11 cryptic sites for vinculin, and it needs to be unfolded for those to be visualized, observed. Okay. So this is the basic process of cell spreading that we've seen lots of times in the fibronectin-coated glass situation. So once activated, it starts to spread. The interesting feature, though, of using the supported bilayers is that now you find when it spreads, there's an accumulation of the RGD and the integrin at the outside of this lipid C here. These are chymographs taking this bar from the first image and that same bar from subsequent images over time. So you can see here that the cell edge is moving out and then moving back in, and as a result of the contraction, initial spreading and then contraction. And so during that spreading, the RGDs accumulate on the outside, and then they move to the inside aspect of that lipid C because of the contraction. So polymerization is pushing them out, contraction is pulling them in, and the surprising thing about Paxillin, is that it's getting pushed out here initially, and then when it gets pulled in, it's able to jump over the barriers because it's cytoplasmic and not so strongly attached to the integrins under the circumstances.
Does it occur only on the leading edge, or like isotropic on the other side?
Pretty much isotropic. Here, the next slide really sort of addresses that question, because here we plot the position, radial position of all of these clusters, and we can see that virtually all of the clusters are moving out. This is the yellow line is the cell edge. The cell edge moves out a little bit further than the last cluster. So the distance between the clusters and the leading edge increases toward the end, presumably as a function of the increased actin polymerization from the edge, which will occur there primarily at later times. And then you see the contraction and everything moves in. Okay. So this is in a case where that 20% or so that will spread on the supported bilayers without the lines. Okay. If we now inhibit Sarc family kinases with the inhibitor PP2, we find that we don't get the accumulation of actin, we don't get the spreading, or the contraction that follows. And indeed, in many previous experiments, we've shown that inhibition of Sarc family kinases is sufficient to block the spreading process altogether. Okay. So as a result of using the supported bilayers now, we see that the process of spreading is a little bit more complicated. First of all, you have a cluster formation. Basically, 3 or more integrins need to be pulled together within 60 nanometers of one another. The Sarc family kinase we've identified as FIN, which is necessary to be active for actin polymerization through the formin FHOD1. Once you get that actin polymerization, there's a local contraction to activate additional actin polymerization, which causes the cell to spread out to a much larger area on that surface. And then you have contraction that's activated by a pressure pulse or tension pulse within the plane of the plasma membrane. Okay. So that the surface area of the cell stays constant during that spreading process until such point as it runs out of membrane and you get a pressure pulse as a result of the actin polymerizing against that membrane, and then you activate contraction. Okay. We think that's a row-dependent process, but have not identified the exact mechanism yet on how that occurs. And then you have this rigidity sensing, presumably. And the reason that we talk about it as rigidity sensing, because when we looked at this, this is now fibronectin concentration here is constant, and this is now soft, intermediate, or stiff polyacrylamide. And we see that when the cells are spread on the soft, they stop at the point where that contraction is activated. And what you find is that there's a rapid flow of actin inward, no adhesions are generated, and whereas on stiff surfaces you find that you see these periodic contractions, the cells spread further and build adhesions on those surfaces. On intermediate, you get the interesting behavior that they try to go out further, the adhesions are not stable, and as a result you get ruffles or upward bending of the membrane or the actins supported by the membrane, support of the membrane, and as a result you revert back to earlier or more central adhesions. Okay. Any questions about this?
The force that is required for the cooling. What are the forces required for cooling?
The forces are generated by myosin and are typically, when measured using pillars, are on the order of several nanonewtons per micron of the edge. Okay, we'll come back to that more in a moment. Okay, because taking a clue from the earlier studies of Chris, which were built upon our earlier studies of using a buried force sensor, it's good to use something on a lever arm to measure the forces on the substrate. Okay. And the pillar arrays are the simplest way of doing that. I argue that this is much better than traction force microscopy. Because even with the continuous nature of the surface there, if you look at the surfaces within the body, they are typically not continuous. Okay, they at least have architectures with features in the nanometer level, and we can now fabricate structures in the nanometer level. So Sabagasemi, a graduate student with Jim Hone, who's a mechanical engineer at Columbia, set about to develop 9 different arrays of pillars, all with a standard geometry, namely that the spacing was the same as the diameter of the pillars, such that the center-to-center distance between the pillars was twice the diameter of the pillars. So a constant fraction of the surface area of the cell was interacting with those, and that now they were made in 3 different diameters, 2, 1, and 1/2 micron, and at each diameter 3 different heights, which had roughly the same spring constant, namely in the range of 2 to 20 some nanonewtons per micron of displacement of the pillars. When cells spread on these surfaces, we got dramatically different behaviors. And I'm running over a little bit here, so interrupt with questions as we go. Okay. So, and the important difference here between the 2 and the 1/2 micron pillars was that there appeared to be roughly constant force per unit area on the large pillars, i.e. that the soft ones were displaced much more than the hard ones. In contrast, on the 1/2 micron pillars, they were all pulled to the same amount and for roughly the same period, consistent with the earlier findings of Benoit Ledoux's lab, in which they varied the substrate rigidity or pillar rigidity by 100-fold and found that they were constantly displaced to a constant displacement. Okay. So that rigidity sensing seemed to involve pulling to a constant distance. Okay. So what was happening over here? Well, when we thought about it, the concept was that maybe the cells were sensing the rigidity of the pillar top, which is PDMS, and has a very high rigidity. Okay. So, one other point I want to make here is that if you now vary the overall rigidity of the pillars, we find that the force per unit area on the 1/2 micron pillars scales roughly linearly over a wide range, and we were getting values up to about 20 nanonewtons per micron squared. Correcting now, after correcting for the bending of the substrate from a study that came out of Viola Vogel's lab, whereas with the 1 and 2 micron pillars, we were working at a constant force per unit area, much lower, at least for the rigid surfaces. Now, when we looked with Paxillin, we found that it was at the edges of the 2 and the 1 micron pillars, whereas it was on top of the 1/2 micron pillars. And the morphology of the cells on the 1/2 micron pillars and the morphology of the adhesions on the 1/2 micron pillars was similar to that on the continuous surface, indicating that what was happening was perhaps, that I suggested was happening, was indeed true, that the cells were sensing the rigidity of the pillar tops. That was even more evident when we looked at the distribution of phosphorylated myosin, i.e. myosin that was activated. We found that it was arrayed around the large pillars, but was between the small pillars and appeared to be pulling the small pillars together. Whereas it was forming a contractile ring around the large pillars here, and thereby sensing the rigidity of the pillar tops. Okay. Any questions about this? Yes?
So then for the large pillars, shouldn't you get the same results for all the different stiffnesses? Because the stiffnesses were only for the height changes, right?
Right. So the stiffness,
diameter dependent.
right, so what we got with that, going back here, is that we have a roughly constant coupling of those adhesions to this actin flow. Okay. So in all cases we have a flow of actin inward, and on top of that, in this lower layer of actin, we have a contraction. Okay. So two processes are going on. Remember, we drew that initially as two layers of actin in the leading edge. That's supported by Jiawei Zhang's results. We're using super resolution. And indeed, we're seeing it again here in that the adhesions form around these large pillars. And what happens when you form those adhesions will couple to that flow of actin inward. Okay. And that's roughly constant per unit area, so that this now explains that. Okay. Yeah.
It looks like from that image that your heights are on the order of the diameter of your pillars.
In some cases, yes.
So how do you calculate the stiffness?
From the bulk properties of the PDMS.
Okay.
Okay. What we should do, and I keep arguing for that, is put a magnetic bead on the tops of the pillars and pull them, and then measure the position from below. But yeah, I mean, that's experiment to be yet to be done. The other issue that complicates this is the potential for artifacts due to the lensing effect of cytoplasm of the cell. We think that that's not a problem because we now go back and use fluorescent topped pillars, and we can measure independent of the, without DIC, the position of the pillars, and we get the same value of 70 nanometer displacement in that experiment. Okay. And we're going to go back and do it again with a polymer which is refractive index matched to water, so that the pillars don't have any refraction, and so we can see hopefully that behavior again. Yeah.
Do you mix different size of pillars within the same substrate? You have different diameters.
We have not. Benoit's lab has been doing a lot of that to try to address the issue of a haptotaxis, movement to different rigidity surfaces. Okay. So, consistent with what I was talking about a moment ago, indeed, when we look at the vectors of displacement of the large pillars, they are all in the direction of the actin flow, i.e., from the edge toward the center of the cell. Whereas if we look at the half-micron pillars, we see these local contraction units that are typically 2 to 3 microns in length. Okay. And we can further now look at this in the periodic contraction situation, and what we find there is that there's a row of contractions which are occurring toward each other, and what we find is that the pillars right near the edge are being pulled inward, and those back from the edge are being pulled forward. And what you should note here is that the ones being pulled forward are being pulled on average about 30 nanometers, whereas those that are being pulled rearward are being pulled at about 60 nanometers, indicating that there is indeed a coupling to that generalized actin flow, and as you would expect, then the pillars at the edge are being pulled more because they're coupled to that overall actin flow than those that are back and being pulled forward. What this also emphasizes is that the forces generated by these local contractions are greater and can be much greater than the forces generated by that actin flow. Okay. And that the forces now we see in these local contraction units are in the tens of nanonewtons per micron squared, i.e. as great as has been reported for adhesions. If we now ask the question of what is anchoring the actin to these sites or to the pillars, we see that alpha-actinin correlates both in time and in position with the contractions. And we see that here building up on the side of the central portion here of the adhesions near the edge, and the ones back from the edge, at least the early ones back from the edge, it's built up on this side, presumably because it was there initially, and as the edge moved out, it stayed there, and now you see this contraction, and you see the myosin is forming in between here. This is done with super resolution imaging using a 3B analysis protocol that was published last year. And so it seems that everything is consistent with this idea that you have actin polymerization from the adhesion sites. You then anchor it now with alpha-actinin, and you pull on it with myosin, and you pull to a constant value of 60 nanometers, and the rigidity signal then is proportional to the force. It may be a digital system where you simply measure the number of these contractile units, and that is the trigger, and that there may be just i.e. a unit force, something unfolds, and if it unfolds, it sends the rigidity signal, and the rigidity signal now causes an adhesion to be built on this site. Okay. So this harks back to what we addressed this question earlier in a study to look at vinculin, GFP vinculin assembly at beads. And we found that with 1 micron beads, you needed to apply a force with the laser tweezers before vinculin would assemble at those sites. Okay. The contact area is 0.2 of a micron, 200 nanometers. And we asked the question at the time, or at least Kathy Galbraith asked that question, of how large a bead surface do you need before the cell will assemble adhesions at that bead. Okay. Because obviously the bead could be likened to a glass surface, and we know that the cell is capable of assembling adhesions at the glass surface. So really, how large an area of contact do you need? And she determined that greater than 3 micron beads would have a sufficient area of contact with that surface that you could assemble adhesions there. And the explanation given at the time was that the myosin filament is 300 nanometers long, and that a myosin filament therefore could not generate force on a 1 micron bead because the area of contact was smaller than the myosin filament, and you needed a larger area of contact. And in fact, if you look at these images back here, you see that the active myosin heads are separated by about 300 nanometers here. Okay. These are 500 nanometer pillars, and this is a good fraction of that. So indeed, it seems that what's happening here is that you require the actin to be anchored by a distance greater than the length of the myosin filaments. Now, another question that is begged by all of this was addressed in the group in Singapore. They varied the distance between the pillars in the study that Sabagasemi did, the one at Columbia. The pillars were always displaced by twice the diameter, and we now varied it over there to 3 times, 4 times, or 6 times the diameter, and got the same profile, namely that there was a peak at 60 to 70 nanometers of displacement, another peak at 120. We also looked there at 2 different cell types, human foreskin fibroblasts versus the mouse fibroblasts. And the human foreskin fibroblasts pulled to 120 nanometers, whereas the mouse fibroblasts pulled to 60 to 70. This was done with Israeli human foreskin fibroblasts, so the Israelis were obviously pumped up about this whole thing, and suggesting that their fibroblasts were stronger, at least the foreskin fibroblasts were stronger. So the sarcomeric units are again about 3 microns. If you look at for the large pillar displacements, again you see there's just a local contraction. Now these are displaced by 3 microns, and it falls off very rapidly beyond that distance. The next question is whether this is really involved in rigidity sensing. And from the work with the laser tweezers, we addressed this issue. And some time ago, to work out a biochemical pathway, which involved, it seemed at the time, receptor-like protein tyrosine phosphatase alpha. RPTP alpha is in complex with the alpha V beta 3 integrant at the leading edge. It activates FIN and SARC that we know are important for this process, so that it was not surprising that RPTP alpha minus cells spread to the same area on soft and rigid surfaces. So we went back and asked the question of what happens in this pillar displacement assay, and sure enough, without RPTP alpha, the green lines here, they don't pull to the 60 nanometer displacement of the controls. And if you now express RPTP alpha in the RPTP alpha minus cells, excuse me, you now get the displacement to the same 60 nanometers. So that indeed this pulling to 60 nanometers correlates with the ability of the cells to generate or to sense the rigidity of the surface. So what we're saying here is that the cell is using sarcomeric-like contractions to determine that the surface will pull back and to also sense rigidity. And for rigidity sensing, they're just pulling the sarcomeric unit to a distance of 60 nanometers. That's a molecular distance. It could be sensed by any molecule that will measure the relative position of myosin and actin. And we're looking now for such a molecule. There are a number out there. Cal-Desmond was the first one we looked at. It doesn't correlate, but others might. in this situation. Okay. So we've gotten to the point of assembly of adhesions. On soft surfaces, they don't assemble. On rigid, they do. And then what happens on rigid surfaces? How does the cell now get a signal from that adhesion? And this gets into Talin, last portion of my talk. And Talin has a binding site. It's N-terminus for integrin, and in the firm domain, and it has an actin binding site at its dimerization site out here at the C-terminus, and in between it has 11 cryptic sites, or 10 cryptic sites for vinculin binding. One of those is not cryptic, and vinculin will bind to Talin in the absence of force to a limited extent. But we showed in vitro that simply applying magnetic force, a magnetic bead force to Talin, we could stretch it and expose those vinculin binding helices, which would complex now with the 4-helix bundle in vinculin to form a 5-helix bundle. Crystal structure of this has been known for some time, and that this is now from the steered molecular dynamics studies out of Viola's lab. Similar studies came out of Roger's lab, where indeed the unfolding would expose these sites, at least in silico, and we tested it out in the test tube, and that worked. So that begged the question of whether it was happening in vivo. So we used this construct from Critchley's lab that had GFP at the N-terminus and M-cherry at the C-terminus. It's a dimer, and in vinculin minus cells it restored normal function, and we see an overlap of the red and green here in these cells. And what was the important step for this paper was that Felix Margadant developed a program that would automatically look for pairs of GFP separated by 500 nanometers from others, and there was an M-cherry in that region. The reason for one M-cherry is that M-cherry doesn't fold very well, and so only about half of the M-cherries present are actually fluorescent. Okay, something you have to keep in mind when using these tools. So when we found an M-cherry with 2 GFPs, that defined a vector that had a length and a direction, and we can determine that direction relative to the actin flow. And in the presence of blebistatin, the myosin inhibitor, there was no polarity to that vector, and the peak was at 60 nanometers here, which corresponded very well with the overall length of the molecule in vitro, and indicated that it was not being stretched. However, in cells that were actively pulling, the vector was now in the direction of actin flow, and the first peak in the length histogram is at 90, another one at 150, and a shoulder here at 210, indicating that the molecules were being stretched to typically twice their overall length. Okay. The most interesting aspect of this comes from the dynamics, which is next. As an aside here, if we look at other molecules like focal adhesion kinase or filamin, again labeled in the same way, we find a different different length histograms. This was now work from the Woods Hole physiology course, which we were fortunate enough to participate in last summer. And the students there in a 2-week period were able to recapitulate basically the data that we got in Singapore, plus look at focal adhesion kinase and filament, which gave dramatically different length histograms. So it seems that many molecules are being stretched in a similar way. The Talin stretch appears to be dynamic in that we see it stretching here to 250 nanometers, dropping back to 0, up to 100, up to 100 again, and up to 100. So 4 times in this period of 60 seconds. Okay. If you look at this relative to the dynamics of vinculin in the adhesion, you find that the lifetime in a photobleaching recovery experiment of vinculin in a typical adhesion is on the order of 16 seconds. So the stretch relaxation cycles are on the order of the same lifetime as the vinculin in the adhesion. Okay. Talin has a longer half-life of about 100 seconds, so indeed Talin should be there for the long haul. The surprising result that we got, though, in this study was that when we looked at these adhesions near the edge of the cell, they were green on the outside and red on the inside. The reason that that's surprising is it indicates that all several hundred to several thousand talins in that adhesion are being pulled in concert. This is consistent with the fact that expression of the vinculin or Talin tail region will also, Talin tail will go to adhesions, indicating that there's something now bridging those Talin tails in those adhesions. And we see that, again, the dynamics are occurring there as well as in the single molecules, and that if we look, this is now perpendicular to the membrane. If we look parallel to the membrane, we don't see movements, and if we look at adhesions near the center of the cell, we don't see the movements as well also. The movements are not correlated in adjacent adhesions, and we see this one being pulled 5 times in 60 seconds, consistent with the behavior I talked about before. But this larger one is being pulled, certainly not showing the same peaks and showing a different frequency of contraction. Again, the controls work. Myosin inhibition brings us down to the background levels. And an interesting control is that the active vinculin head, which it binds essentially irreversibly to Talin now and causes elongation of adhesions and stability of adhesions, it causes a increased stretch of Talin to about 400 nanometers in this case, in some cases up to 600 nanometers. The overall length that we could expect for the Talin molecules are roughly 1,000 nanometers, so that it's probably a significant fraction of the overall length is being stretched. So the model that we come up with here is really a stick-slip model in which the actin is flowing inward. It sticks or attaches to the C-terminal portion or aggregate of the talins, causes them to stretch, exposing now vinculin binding sites. Vinculin binds, and then this slips. And the reason it slips is that the overall length, as I say, is going to be maximally 1,000 nanometers. The flow of actin here relative to this site is roughly 60 nanometers per second. The lifetime of the vinculin or the Talin in the adhesion is 100 seconds, so that works out to 6,000 nanometers of actin flow, so it can't stay attached. Okay, it's got to break. And when it breaks now, we see increased release of vinculin, And so one way of thinking about this is that maybe the signal that comes from this cycle of stretch and relaxation is indeed a phosphorylated or modified form of vinculin that was modified while in complex with the talin. So this is a way that the cell can integrate over time because it depends upon the number of adhesions, the frequency of actin flow past those, and as well as the dephosphorylation or phosphatase activity within the cell. So you've got a nice way of regulating and integrating over time. Certainly cycles are good integration tools, and this has been talked about in a number of other cell systems. The example that works very well is to consider what happens with the calcium calmodulin kinase. Calcium calmodulin, 12 calcium calmodulins bind to that kinase to fully activate it. Okay. It's present in synapses, and it's an important component in the stabilization of synapses. So conceptually then, you have a depolarization at a synapse, you have calcium entry, you have now the binding of 1 or 2 calcium calmodulins to the kinase, and if you don't have any further firing and calcium entry, those can come off because the calcium level is very low in the non-firing situation. But if you have repeated firings, you can build up the complex to the 12 calcium calmodulins bound to a single kinase, and that sends a signal back to the cell body, and you get consolidation of that synapse. So that's an integration method through a quote-unquote feudal cycle, the feudal cycle being calcium entry and exit. And the same we're thinking of in terms of, or analogous process, in terms of this stretch relaxation. So that the stretching of proteins is not going to be sensitive to a simple bump like this, but indeed to the multiple cycles of stretching and relaxation that would occur over time. And allow that integration that, as I mentioned before, is critical for understanding the biological processes, i.e. how those two identical twins can look the same dependent upon stochastic processes. So what I've tried to tell you today is that the process of adhesion formation involves a series of if-then questions that the cell asks of that substrate. Okay. And dependent upon the answers to those questions, you will or will not develop adhesions, and then you will test those adhesions or develop a signal from those adhesions through the stretching and relaxation of talin. Similar processes are going to occur in a variety of other different types of motility and motile processes, and the reason that we're building this MB Info site is because those processes are essentially the same for many different cell types. And once we can build this around function, a functional database, we don't have to do it again, okay? Because all those functions will be defined. It's a wiki site, so we appreciate feedback on this. And the site is meant as an educational as well as a research tool. And we have lots of diagrams and animations, etc. there. The topics that are present on there are lamellipodia, motility, filopodia, podosomes, invatopodia, cell matrix interactions. and cell interactions are coming soon, as well as nuclear functions. And I wanted to re-emphasize again that local contractile units, 1 to 3 microns in length, analogous to sarcomeres, appear to be important not only for developing muscle, stress fibers, but also for sensing rigidity at the leading edge of the cell, and that those can then generate signals going to the nucleus dependent upon the matrix environment that the cell is in and it's the rigidity of that. And give credit where credit is due. The supported bilayer studies were done by Cheng-Han Yu. Mark Schwartzman, working with Shalom Wind, was responsible for the basic adhesion unit. Armando Del Rio did the in vitro stretching of Talin. Felix Margadant, Lily Chu, and Edna Hugh, the in vivo studies. Thomas Iskrach, working with Cheng-Han, defined the formins that were important for assembly there, and Saba Gassemi, working with Jim Hone, did the pillar studies. And the pillar studies in Singapore were done by Feroz Mustafa. I thank you very much.
What I'd suggest is that we take the break.
Okay.
Mike, you'll be around for
Right, for lunch.
through lunch, so everybody should have a